Treponema pallidum. In this session, we will talk about this spirochete and also about the biosafety lab practices regarding Treponema pallidum. Now, Treponema pallidum is an example of fastidious spirochete as it is helical in shape. Spirochetes or Treponema pallidum are motile as they possess flagella. Treponema pallidum is of great medical importance because it causes syphilis, penta, yaws, as well as Bagel. If we look at the morphology of Treponema, then it has a rich cytoplasm containing genome, unbound membrane organelles like all other spirochetes, as well as 70S ribosomes. If we look at its outer membrane, it is lipid-rich in nature and it is of great serological importance. Now, spirochetes or Treponema pallidum are actually gram-negative bacteria and they should appear pink in color after gram staining technique. But Treponema pallidum is so small and so thin that they cannot be examined under light microscope after performing gram staining technique. So the only way to examine Treponema pallidum is by using dark field illumination technique, in which Treponema pallidum become visible against a dark background as they illuminate in the darkness. Now the interesting thing is that Treponema die upon desiccation, that is in dryness, and they also dry in the presence of atmospheric levels of oxygen. Now Treponema pallidum can be diagnosed serologically, but they cannot be cultured in vitro microbiologically up till now. Now Treponema pallidum causes syphilis, but its subspecies such as Treponema pallidum endemicum causes Bagel or endemic syphilis. Second subspecies Treponema pallidum caritium causes Pinta, and the third subspecies Treponema pallidum pertuni causes Jaws. If we look at the transmission route of Treponema pallidum, then it is a sexually transmitted disease and it is acquired through close sexual contact. Treponema pallidum has the ability to enter into the host via penetrating or breaching two types of epithelium. One is the squamous epithelium. For example, it is present in mouth, and the other type of epithelium is known as columnar epithelium, which lines the respiratory tract. Now, Treponema pallidum can be transmitted from the mother to the fetus during the later stages of pregnancy and the infant born with congenital syphilis. Either the dead baby will born, or if the baby born with congenital syphilis suffer from mental retardation. Treponema pallidum is a spirochete, so it has a helical structure. But if we look at its mortality, then it is quite interesting because it moves in a corkscrew motion in a viscous medium. For example, in mucus, when in the body, when it passes through the mucus, it shows its corkscrew motion. Now let's look at the genome of Treponema pallidum. Treponema pallidum is one of the smallest bacteria and it is 1.14 million base pairs. If we look at its metabolism, then it has limited metabolic capabilities. And if we look at its three-dimensional structure through bioinformatics, then it appears flat or wavy in shape. For the identification of Treponema pallidum, we use special types of strains such as Dytrus strain. Serologically, we use two types of methods. One is the agglutination technique, in which we make use of carbon particles to make syphilis visible against a white background. Apart from this, enzyme-linked immunosorbent assay can also be used for the diagnosis of syphilis as it has greater specificity and sensitivity. Unfortunately, no vaccines have been developed against syphilis, but the outer membrane has few proteins that helps in the identification of bacteria serologically, because these proteins are for an antibody that is more effective way of diagnosing syphilis. Now keep in mind, if we look at the biosafety lab practices of Treponema pallidum, then there are primary hazards that it can be transmitted to lab personnel handling the serum of Treponema pallidum or serum samples containing syphilis. Now there are two ways by which the syphilis can be transmitted to the lab personnel, either by accidental parental inoculation or in other ways when there is any wound, injury, the Treponema pallidum can penetrate the infectious or the broken skin. Apart from this, Treponema pallidum, whenever comes in contact with the mucous membrane, it can infect the personnel or the lab workers.